This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 526, recorded on December 21st, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Who I just saw earlier this week.
Yes, we had a neat time and out on the campus of the veterinary school, and that'll be a TWiV coming up sometime in the new year.
So yesterday I did a TWiV with Elio Schechter, and he was at Tufts Medical School, and he said he was involved in setting up the veterinary school.
Oh, cool.
In the 70s. How about that? He knew all about it. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
I feel like I've spoken to you lately.
Yeah.
I guess we did.
Yes.
It's so weird to speak outside of TWiV, isn't it?
It is.
Yeah.
And from Austin, Texas, Rich Condit.
Hi, y'all.
Y'all.
All y'all.
All y'all.
Yeah. Yes.
I don't know. It's only been a week, but I feel like I haven't spoken to you in a long time. Why is that? Because we're all busy and shit happens, I guess.
Yeah, something like that. But it has only been a week.
This is my sixth podcast of the week.
Well, that probably explains a lot.
Yeah.
What's wrong with me? So it was Monday, Tuesday. It's Monday at Tufts. Tuesday at Microbiotics, which is Islam Hussein, where he works, little company in Worcester. And then yesterday I did 3 Immune, TWiM, and TWiVo.
Wow.
Today TWiV, only because of the vagaries of scheduling. I don't want to do 3 in one day. Although I do enjoy it.
Well, there you go.
The 3 yesterday are still in the can. I haven't had a chance to put them up. But Nell said, I admire it. And I said, don't admire it. There are other things I should be doing. Always. In an effort to make ASM's podcast the best they can be, This Week in Virology wants to learn more about you, the listeners of TWiV. Please take 5 minutes to answer a short 12-question survey. Surveys like this help us to collect data needed to seek sponsorship, which would allow us to grow the podcast. You can find the survey at asm.org/TWiVpoll. That's asm.org/TWiVpoll. Thank you for your time in listening, subscribing, and helping us improve the show. We appreciate the support. You may remember that we did a poll about 6 years ago. 1,000 of you participated.
I hope we get good participation this time.
Please do it.
I'd really like to see the results of this.
Yeah, I'm sure it's changed. And we're doing this for TWiM as well. And I've been doing it for 3 weeks. We have only 75 respondents. But here on TWiV, we can motivate you.
Did you tweet it?
No, I did not.
Because I was going to say, if you did tweet it, I missed it.
I didn't tweet it. It's a good idea, though. Because, people are listening in their car mostly, and they can't do a poll.
Right.
So I have a feeling more and more people are listening in situations where they can't do other things. Mm-hmm. So, yeah, it's a good idea. I should tweet it.
Got their hands full of plaque assays.
Or they're steering a car. And they don't want to run into an embankment.
Right.
Or an abutment.
Is that what he said, an abutment?
I think it was an abutment.
So. Yeah.
Okay, Kathy, what is up with ASV?
Okay, American Society for Virology annual meeting will be at the University of Minnesota, July 20th to 24th, 2019. So this is the first day of winter, and soon, February 1st, will be the abstract deadline. And if you want to apply for a travel award, you need to become a member. If you need a sponsor for your abstract, they need to be a current dues paid full member. All of that needs to happen before February 1st. So now, while you have some time, think about becoming a member or paying your dues or whatever you need to do while you're thinking about the scientific content of your abstract. You can do these other things. I want to highlight one thing that's kind of unique for this year, and that's one of the satellites, is going to be about having a career that's at a college or university where it's teaching focused. And so there's a whole lineup of really good speakers that the Education and Careers co-chairs put in for this satellite. It includes, as I said, a number of people, including Breanne Barker.
Woohoo! Excellent.
Yeah. So the cool thing about this is that, of course, it's going to be only open to trainees, postdocs, students, and for them it will be free. But because of that, we have to limit it to the first 100 people. So obviously, if you're thinking about having a career at a teaching institution, this satellite will be really good. The program for it looks terrific. And so consider registering for that satellite. That's on Saturday before the main meeting begins.
Is that program available online?
That is a good question. The poster is, and Minnesota was hoping to have their content for that program on the site, and I just haven't checked it lately. So
I'll do so right now.
Yeah. So for the membership for ASV, that's asv.org, and it's a slightly more complicated one for Minnesota that I haven't memorized yet. And then it switches to another URL. Oh, good.
It's here.
Good. I hadn't looked yet. All right.
That's the meeting that you've pasted. We'll put the meeting, the summary of the meeting, symposium, and other things in the show notes if you want to see University of Minnesota.
Right. This is just the link to the satellite, but one up from that is the program itself.
Mm-hmm.
Yeah. Yes.
Howard Temin's legacy. Cool. Did I tell you? I might have, but I'll tell you again. I was giving a talk at University of Wisconsin-Madison. This has to do with Howard Temin. And just go through a little of my history. I said I worked with David Baltimore. I went to his lab shortly after he won the Nobel Prize for discovering reverse transcriptase, and I continued. And then someone came up to me afterwards and said, this is Madison. You have to mention Howard Temin as a co-discoverer. Okay, I will next time.
Yes.
And nowhere else. Yeah, Lou Man is good at giving everyone credit. Guess I'm not.
That's quite a cultivated skill, and it's really, as one of our writers pointed out, a nice skill to have.
Yeah,
you have to work on it. I remember one time after a lab meeting, I basically attributed discoveries from a 2-author paper, where the senior author was a male and the first author was a female postdoc. I referred entirely to the male senior author. And afterwards, Joan Stites gave me a little lesson in attribution.
Good.
She was displeased.
And you've learned your lesson.
Oh, yeah. That was a good one.
As everyone may know, or some of you may not know, or some of you may know, in all possible permutations of that. I'm doing a crowdfunding to try and raise money for the lab. We are at 28% of our goal of $15,000 with 10 days to go. Crowdfund.columbia.edu. And as part of the rewards, we have a shout out for some of the donors, and here are just a few. And interestingly, they write curious things in their little dedication. So Sal Baptist wrote, in honor of Dee, the polio survivor who zooms around Highland Park, New Jersey in her scooter. Peter Brandano, in memory of my mother, Joyce Brandano, who suffered with polio as a child. Marcy Ritter, in memory of Constance Armitage Wallace. Carol Shoshkis-Reese, in honor of Vincent Racaniello. I'm glad it's not in memory of Vincent Racaniello. Valerie Cusick, A.V. Phlox, Gertrude Ray, Andrea Agree, Ruth Perez, Marion Weiss, Alicia Steinhardt, in memory of Dr. Richard Steinhardt, class of 1966. Maddie Corper, Met Werdelin, Kisandra Metusik, Alan Russo, Daphne Cooper, Michaela Kavanaugh Cohn, Justin Levitt, in honor of the phages of the world. Sean Gernant, Rachel Scott, LeMay Axton, in honor of my beautiful little Cambria Annabella, victim to AFM, September 23rd, 2016, quadriplegic.
Wow.
That's tough. So AFM can last. It can remain always.
Right.
It doesn't always resolve. So a lot of these individuals have problems. So I appreciate their support. We have 2 follow-ups. Sue Ellen writes, TWiV 525 was the bee's knees. Another excellent podcast. You're really very, very good. Learned a ton about bees. Have listened twice. Might give it a third listen. Go for it. Alan.
Andrea writes, Dear TWiV team, thank you for your episode featuring Dr. Flanagan and her work on honeybees and their viruses. As a virologist trained in entomology, a backyard beekeeper, and an avid gardener, this story landed on the pistil of the flower-shaped Venn diagram of my personal interests. I shared the podcast episode and one of the MSU educational videos with other bee enthusiasts at our local Beekeepers Association. We're always looking for reliable resources to educate ourselves and the public about the importance of pollinators and the challenges they face. Thanks for all this wonderful new material. And that's from Andrea, who is a research assistant professor at Vanderbilt, who sounds like somebody we might want to contact. It's
interesting. These are all hobbies. Her position is in pediatric infectious diseases.
Right, but a virologist trained in entomology got my attention.
Yeah. I know Andrea.
Okay.
In fact, her first name is really something else.
Yeah.
That's close, but she calls herself Andrea because it's easier for everybody.
Yeah, I just looked up her page and it has the second name in a parens. Yep.
Mm-hmm.
I wanted to talk briefly about Aaron Klug, who passed away not too long ago.
The age of 92.
92. And I would like to reach 92. That's neither here nor there.
I'm going for well over 100.
Okay, do it.
I got a Christmas card from somebody whose mother is 102 or 3. Yeah.
Wow. Well, Aaron Klug...
92 is not bad, though. That's
yeah.
Aaron Klug was a crystallographer, and he won a Nobel Prize for important insights into 3-dimensional structures of viruses. And I particularly wanted to briefly talk about him because he features in my virology lectures. He was very important for how we understand icosahedral structures of viruses now. And there's an obituary in Nature, which is quite nice. And I really like the point where he moved to London in 1953 because he had been denied a U.S. visa because they said he belonged to a student organization that the South African government deemed communist, and McCarthyism was raging at the time. So he moved to London and he met Rosalind Franklin on a staircase. That's so cool.
Yeah.
And it says it was an epiphany. His theoretical skills combined perfectly with her technical skills. So they collaborated and solved the structure of tobacco mosaic virus. And then he worked with Donald Casper to try and figure out some of the broader principles of icosahedral symmetry. So, as many of you may know or should know if you've listened, many spherical viruses are built with what we call icosahedral symmetry, where you can have 60 subunits, like a protein subunit, and they can be a single protein repeated 60 times. That would be a very simple icosahedral packing strategy. And in that kind of environment, all the subunits interact with their neighbors in an identical or equivalent manner. All right. That's the simplest kind of particles. And Watson actually and Crick had worked on understanding those previously. But we knew already by the time Casper and Klug were working on this problem that there are bigger viruses, right? And since viral proteins don't get bigger, how do you make a bigger virus? They're all about the same size. And what you do is you add subunits. And you can add more than 60, right? Like 180 and 240 and many, many more as the viruses get bigger. And so what Casper and Klug did is developed a framework to explain how this works. And they started with the concept of triangulation, which is each icosahedron is made up of triangular faces, icosahedral triangular faces. And they divided into smaller triangles or facets, and that facet number is T. triangulation number, the number of structural units or small triangles per face. So you can always figure out the number of subunits in a virus particle by multiplying the T number by 60. So, T equals 1, 2, 3, 4, or whatever. Well, not every T number is allowed, but you can multiply it by 60 to get the number of subunits. Now, the key that Casper and Klug developed was the idea of quasi-equivalence. That is, things are similar but not the same.
Right.
Quasi-equivalence. So when you put more than 60 subunits in a capsid, you now have different kinds of interactions. They're generally similar, but you now have pentamers and hexamers. So in the 60 subunit virus particle, you only have pentamers. But now when you start putting more in and the particle gets bigger, you now have pentamers and hexamers. So if you think about it, there's got to be some slightly different interactions. And that's the theory of quasi-equivalence and triangulation number. Those are the two key contributions of Casper and Klug, which I love explaining to students in my course. And then they tell me later it was their least favorite lecture. I think it's just great how it makes sense. For me, it's cool because it makes sense structurally and numerically, but they don't care. They want to hear things that are very cool. Now, here's one more thing I want to tell you. We now know, in this Nature article, they kind of summarize this, and they say all known spherical viruses can be classified in this way. No, it's not really true. We now know that there are a number of viruses that depart from the constraints that are imposed by quasi-equivalence, like polyomavirus, simian virus 40, has a T equals 6 triangulation number, which Casper and Klug excluded in their rules. They say we can't have a T equals 6, but we do have viruses that have that and others as well. So whoever wrote the obituary, you should watch my lecture. And next time, I don't know if Casper is alive. Does anyone know? Donald Casper. American structural biologist. He's still alive. He's 91 years old. Wow.
Media uses present tense. It's usually a good indicator.
Yes. Usually they put the year when you've died. He's still alive. An American structural biologist. So they collaborated. Well, if you're going to write his obituary, just get it right next time. Okay? Then I'm sorry that it was too long, Kathy, but just summarize.
No, you did summarize it quite a bit.
I like to write it all down. That makes me sort of
Yes.
put it in my head, right? I don't ever mean to read it, of course. But and I put a picture in, which I'll put in the show notes, of T equals 1 and T equals 3, so you can see how T equals 1, all the subunits interact the same, and T equals 3, they're similar, but not quite identical. Quasi-equivalence.
I have to say, you do a good job of it. And I tried to do it a couple times in my class. And I think either the first or second time I did it, I totally succeeded. And then and Beth, who I co-teach with, said, that was amazing. I even understood it. And then the next year, it totally fell flat. And I just said, I'm not going to try and do that again. So
it's interesting. In the beginning, I had problems with it because I hadn't done it enough, I guess. And then it got better as I went on.
Do you use models? Do you have any models you can use when you're talking about this?
I do, but I think the pictures are good also. But a model would be great. I don't actually have T equals 1 and T equals 3, but I did make this last year, the buckyball video, where I made a T equals 1 and a T equals 3 capsid with magnetic buckyballs. And I made each pentamer a different color, so you could really see that it's all pentamers in the T equals 1. And then in the T equals 1, you can see pentamers and hexamers. So I think the buckyballs really make it come through clearly. So, all right. Thank you, Casper and Klug.
Yes.
And I have a snippet, which is our papers are both from MBio today, interestingly.
Yeah.
It just happened that way.
And therefore open access.
Yep.
Yeah, they're open access. Our snippet is all about prions. Prion seeds distribute throughout the eyes of sporadic Creutzfeldt-Jacob disease patients. And this comes from the NIH, University of California, San Diego, UCSF, the National Institute of Neurology in Havana, Cuba, and Case Western Reserve University. And first author is Christina Oru, and the last author is Christina Sigurdsson, which is really interesting because there was a guy named Sigurdsson with 2 S's years ago who wrote a lot about slow virus infections. And I'm sure she's not related because she's got 1 S, but it's just kind of interesting. She's the senior author. Also on this paper is Byron Caughey. He's from Rocky Mountain Laboratories in Hamilton, which is part of NIH, and he was on a TWiV a while ago. So as everyone may know, we talked about prions on this show before. These are proteins that can be transmitted or in some cases misfold spontaneously within you and cause neurodegenerative disorders. And we can't treat them, so mostly you die. You can acquire them by ingesting contaminated food or by getting human products like blood products or a corneal transplant, as we'll see.
Or eating brains.
Eating brains, yes, you shouldn't do that. Or eating hamburger meat from cows with bovine spongiform encephalopathy. Or it can just happen because you've inherited a mutation that predisposes your prion protein, PRP, to misfold. And the idea is that, let's say you ingest some contaminating prions in a meal. They make their way to your CNS, and that's a big step.
Yes.
How that happens, we're not sure. And there they encounter your prion protein, which is properly folded, and because they're misfolded, they will make it misfold, and goes on and on and on until you die of the disease.
And these are not viruses, but they've ended up being studied by virologists because virologists find this interesting.
Absolutely. As some people say, when these theories first arose, only the virologists would listen.
Right.
So now they're in the virology textbooks.
It's a very simple system that replicates itself and does so without any traditional genetic material. And to a virologist, that's like, wow, even simpler than viruses. I love it.
Mm-hmm. So
that wasn't the initial reaction. He got Stanley Prusner, right?
Yeah, Stanley
got a lot of flack for that.
He did. Mm-hmm. So Creutzfeldt-Jacob is one of the names for this human disease. There are others because there are different kinds depending on the pathology and the incubation and all that. There are also prion diseases of animals. Chronic wasting disease of cervids is one, but there are of mink and others as well, and cows, of course. Sheep is the classic one, scrapie. So in people, they can be transmitted from prion-contaminated material, and that's what this paper is about. So visual disturbances are an early symptom of Creutzfeldt-Jacob. I think this says here there are 10 to 20% of the cases you have eye problems, and then in late stage, blindness develops in 25% of these patients. And corneal transplantation has been shown to transmit the disease. So, for example, you would die, and it's not known that you have Creutzfeldt-Jacob, but the prions are in the cornea, and then it's transplanted to someone else, and then they get it. There have been a number of reports of that. And there are a lot of corneal transplants done. 185,000 in 2012.
Yeah.
Worldwide. So we need to, so the purpose of this paper is to figure out, how often are there prions in the eye of patients with CJD.
And where in the eye.
Yeah. So there's a cool assay that's been developed in which we have talked about before, the real-time quaking-induced conversion assay.
Love the name.
RT-Quick. It's kind of like a PCR for proteins. Right? And you take a sample that you think may have some. So when the prions misfold, they tend to oligomerize, right? And so you take a sample that you think has misfolded prions in it, and you add some normal monomers, and you incubate them, and the monomers will be converted and become oligomers. So that's the incubation part. And then you sonicate these long oligomers and break them up into smaller bits and repeat the incubation. And so each of the smaller bits will then grow. And then you sonicate and break them up and let them grow again. And so after a number of cycles, you can find these proteins like on a Western blot or by some other means.
It's not always sonication, right? Sometimes they're just put on a shaker table.
Yeah.
Right,
Yeah,
that's what they do in this paper.
Yeah, and it's shaking at like 700 RPMs. So it's
Is
really shaking.
Really shaking, yes.
And
yeah.
happily, the oligomers bind a fluorescent dye.
Mm-hmm.
Right.
So the amount, or a dye that I guess fluoresces when it's bound.
Yeah.
At any rate, you can follow the oligomerization just by following the fluorescence.
Yeah.
So I guess you can do that with a plate reader, right? So you can do all this
in
in
the
a
real-time part of it.
Yeah, you can do all this in a 96 well format. So that's just cool.
Mm-hmm.
Right. So there's this RT Quick is shaking, and there's another version involving sonication, right?
A 96
Right.
well format with tight lids.
Right.
This was originally developed to check out cows because, if you're using a cow for meat, you slaughter it before it has symptoms typically of bovine BSE. And so they said, well, can we detect these? And they found that the prions can be found in the nasal cavity of cows who do have BSE. And this assay was developed in a couple of places, and one of them was over at Rocky Mountain Labs. And it was pretty accurate, quite sensitive. In other words, in people or animals that have these diseases, 100%, approaching 100% sensitivity and specificity.
Right. That's the main breakthrough for this paper, is that they can now go look at samples with this really sensitive assay and look at all these parts of the eye.
Right. And in fact, that was a real breakthrough for the whole field, I think, because previous prion assays involved infecting a susceptible animal, and these things take years to develop, and so people would have experiments running for a whole grad school PhD period, and they'd finally get their result. So this is great. This you run this experiment and you get a result right away.
And for people, previously you had to die, and then they would take brain samples and say, "Oh yes, he or she had prion disease." Well, we couldn't do anything anyway, but this is what you had. And now perhaps they could find out earlier. So if you have neurological symptoms consistent with it, they could do a nasal swab and look. Of course, there's nothing you can do anyway.
Still can't do anything, but this is common to a number of diseases like Alzheimer's, where treatments are not really available, and so people say, well, why bother to have a diagnostic? But you can't develop the treatments if you don't have a diagnostic, so you really need to start there, and this is definitely a step in the right direction. Yeah.
Now I presume more efforts to make drugs will occur.
Yes.
And the other reason for this is that if people have this in their eye and it's not known, that can contaminate instruments, and then there are, not only does it have to be the corneal, can it be the corneal transplant, but it could just be from examination tools.
Yeah, right.
And these things are really hard to get rid of.
Yeah, autoclaving won't get rid of them, right?
Autoclaving doesn't get rid of them. All the stuff that destroys DNA and RNA and kills off all the viruses doesn't get rid of them. You have to, I think you have to phenol swab, and that's not even practical for some of the surgical instruments.
I had an email recently from someone who asked if she could get prions from her shower. I don't know of any case where that happens. You usually have to have contact with contaminated material, and I don't think water is going to do it. So in this study, they analyzed the eyes from 11 cases of individuals who died of pathologically confirmed sporadic Creutzfeldt-Jacob. So sporadic means you just develop it and there's no genetic predisposition, but protein misfolds and that's it.
Right.
Who knows why? And 3 of these 11 had visual disturbances of various sorts. And they, they were diagnosed to have CJD by the quaking and shaking of brain tissue. So they looked in the eye, 8 eye regions. Eye has lots of regions.
Yes.
Muscle, optic nerve, cornea, lens, etc. All 11 eyes were positive in the quaking and shaking.
And positive everywhere.
Everywhere. Yeah. And convincingly positive. You look at the graph. Yeah, I believe it.
Yep.
Mm-hmm.
Big error bars, but it's quite clear that they're positive,
Big number of samples.
And they also did some immuno labeling for the prion protein in the eye to see where the protein was exactly, and they could see exactly where it was in different parts of the eye. But the bottom line here is that this is a great assay, and you could take, so let's say you have a cadaver with corneas that are going to be donated. You could then take other parts of the eye and say, are there prions here?
Right.
And if there are, then you throw the cornea away.
The place to assay, I think, would be the retina, because that's where they found the highest levels.
Yeah. And in the meantime, they keep the cornea on ice, or wherever they keep it.
Right. Right. And so they had kind of a theory about how maybe the prions are coming from the brain to the retina, and then from the retina to the rest of the eye.
Right. So from the brain via the optic nerve, right?
Optic nerve, yeah.
Mm-hmm. Yeah, and so that's it. So if next time you need a corneal transplant, make sure they check it for prions. What would cause you to need a corneal transplant? You could damage, I guess, would be one thing. Oh, like herpes virus infections.
Yeah, there are eye diseases that I think degenerate the cornea.
Mm-hmm. All right.
I'm sure somebody can write in and brief us on that.
Brief us. But the bottom line is we can now tell if your corneas that you're about to receive have prions or not.
And I think at this point, since it's known that the corneas can carry this, there may just be a policy that somebody dies of neurological disease not otherwise specified. You're probably not taking their corneas.
Yeah, but you could imagine someone gets in a car accident and is killed.
Right,
You don't know. They could have Creutzfeldt-Jacob at an early stage, right?
Yes. It is a very, very rare condition. We should probably point that out. But yeah, this is something to watch for.
I want to tell people exactly how rare it is. I think sporadic Creutzfeldt-Jacob, which is most of the Creutzfeldt-Jacob.
Most of the cases, I think.
1,000, fewer than 1,000 cases a year
in the world.
It's a good question. Fewer than 1,000 cases a year in the world, probably. Doesn't say. It just says 1,000. That's unfortunate when they don't say, isn't it?
Right.
Let's click on it. Fewer than 1,000 cases a year. And then the other forms are even rarer. So there you go. Okay. That's our snippet. And that's a true snippet, isn't it?
Yes, we actually kept to a reasonable time.
And now we have a paper.
Which is definitely not a snippet.
It's not. We can chat about this for a while. It's very interesting. It's also in Bio Origins and Evolution of the Global RNA Virome. And the authors are Wolf, Kozlowskis, Aranzo, Lucia Sanz, Jens Kuhn, Mart Krupovich, who I had invited to ASV a number of years ago, Valerian Dolja, and Eugene Koonin, who's been on TWiV. Virocentricity with Eugene Koonin. And this is about figuring out, as Kathy mentioned earlier, the phylogeny of RNA viruses and some speculation on where they came from. So to put it into context, we think that the first life that evolved were RNA molecules that developed interesting catalytic activities. Eventually DNA emerged. Viruses that have RNA are kind of relics of this old RNA world.
And in fact, as they point out in the paper, the first things that could be classified as life, the first self-replicating life forms, were quite likely RNA molecules that behaved a lot like RNA viruses.
Yeah.
So before there was anything to parasitize, there were viruses.
In an otherwise acellular world, right?
That's right. That's right.
The primordial soup.
Right. That's right. So, not a virus as we call them today, because we always say require a cell, but it's a self-replicating entity. It's close. And so
then I guess at that point we couldn't call it life, could we?
Well, we don't want to get into that.
Yeah, we don't want to go there.
Look, if it's self-replicating, it's pretty good, no?
Yeah.
Yeah, we're going to have to go there towards the end of this.
So
yes.
Basically, viruses were once living and then they died.
Right.
I'm just joking. So as everyone knows, I've said that now 3 times. I really am sorry. Let me remind you that the Baltimore scheme, there are 7 classes of viral genome, and 3 of them are RNA viruses. The plus-strand RNA viruses, the minus, and the double-stranded RNA. I would add the reverse transcribing plus-strand RNA, but the paper authors did not. So
right, they only count for this. They're saying RNA viruses are viruses that replicate solely with the RNA. Yeah. They do not have a DNA intermediate.
Well, the viruses with RT will come into play here.
Yes, they will.
They will. And
I was deep into this paper. And I got to the figure. I said, wait, where are the retroviruses? Oh, right. No, that's not.
So there are a lot of RNA viruses on the planet, of course. But interestingly, and this will come up later, there are not a lot of RNA viruses of prokaryotes, of bacteria.
Right.
And I think Ben Tenuver mentioned this when he was talking about this. There's only one family of positive strand RNA viruses, the Leviviridae, and the one family of double-stranded RNA viruses, the Cystoviridae, that infect bacteria. You should remember those names, Leviviridae and Cystoviridae. And interestingly, there are no negative-stranded RNA viruses of bacteria that we know of, but maybe they're out there and we haven't found them, right? Could be. They actually acknowledge that. And so there are lots and lots in eukaryotes, because maybe the eukaryotes provide a nice, hospitable, protective environment, and the bacteria do not.
Because they have compartments.
Right.
And the other
membranes,
possibility that they mention is that without a nuclear envelope, DNA viruses could beat out RNA viruses in the bacterial cells. So the bacterial cells don't have a nuclear envelope, and that's another hypothesis they have. Yeah.
Very cool.
The idea being when you put in a nucleus and a nuclear envelope, it's one more barrier that a DNA virus would have to deal with. So it puts it at a disadvantage in the competition with RNA viruses.
So there are lots of RNA viruses globally, and we're learning more and more because people are doing metavirome studies where they just get environmental samples and they just sequence all the RNA viruses in them. So we've accumulated lots of new sequences. Plus, there have been recently lots of invertebrate virus genomes sequenced in various samples. We talked about a paper here on TWiV where they identified over 200 new invertebrate viruses, RNA viruses. And so this has given a whole new range of sequences to look at, and that's what they do in this paper. They take advantage of that.
Right.
In particular, they look at the RNA-dependent RNA polymerase gene, which is the single gene that's universal among all these RNA viruses.
Yeah, so a big problem with classifying the RNA viruses is that they mutate a lot, and there are a lot of them, and they don't have a lot of similarities. But the one thing they have in common is they all need an RNA-dependent RNA polymerase. So
everyone has this, and it's the only thing in common among all these viruses. Right?
And we're going to slip probably and call it RDRP, which is short for RNA-dependent RNA polymerases. And just to point out that for the most part, cells aren't going to have or need these enzymes. The RNA viruses are. They have to be able to make more RNA from RNA. And that's pretty much unique to these viruses.
Now, these RDRPs are part of a bigger class of polymerases called, which have palm catalytic domains, because their structure looks like a hand with a palm domain, and that's the catalytic domain. So the palm domain polymerases, which would include reverse transcriptase and DNA polymerases.
Is anybody not holding up their hand right now?
I am. I'm looking at it.
Yes.
Why am I looking at my hand?
Because you're about to grab an RNA molecule.
Which hand are you looking at?
My right.
Yeah. I think it's the right hand that these enzymes resemble. If Dixon were here, he would say, I resemble that remark. So I say it for him. So these RDRPs, by the way, are highly similar to the reverse transcriptase, a specific reverse transcriptase of retrotransposons in prokaryotes that are thought to be ancient, ancestral to all the reverse transcriptases. So very similar to the RDRPs of plus-strand RNA viruses. And so this is actually a theory that has been around already, that the plus-strand RNA viruses, by the way, which are thought to have arisen first, because they're relatively simple. It's just a plus strand that's translated, right? You don't have to do anything else fancy. They may have arisen from prokaryotic reverse transcriptases. But there are other scenarios, and we didn't have a lot of data up till now. So, yes, sir.
That ancestral organism or ancestral enzyme is missing. We don't have that.
We don't have
it's presumed.
That's right. It's missing. Okay. There are a lot of
rightfully presumed, but missing nevertheless.
Right. And so in this paper, they take advantage, as I said, of the. I love this sentence. Before I get to that, they say the prospects of substantial progress remain dim because of the sequence divergence, as Alan said. But we now have revolutionary developments in metagenomics, so we have lots of new sequences. Plus, we have this invertebrate virome. So that's what they use in this paper. And they collect thousands of sequences of RDRPs and RTs, reverse transcriptases, 4,617 RDRP sequences, and 1,028 reverse transcriptase sequences. And they analyze them using a different approach, which is called iterative clustering alignment phylogeny procedure, which is not simply making phylogenetic trees, but making alignments and going back and checking them and then doing it again. As you might imagine, they have a computational pipeline to do this.
Yeah, I don't know if we mentioned, but this is done at the NCBI, the National Center for, what is it?
Biotechnology Information.
Biotechnology Information, which is, that's what this institute is all about. And, there are other contributors from elsewhere as well, but its home base basically is the NCBI. And I was thinking as we were doing this as well, that this is as basic research gets.
Yes.
And the NCBI is just the fact that we have this thing where this kind of research can be done. Mm-hmm. We have this institute where this kind of research can be done is just marvelous.
Yes.
It's great. And all these,
because otherwise I'm not sure this would get done,
Well, and as we're going to see, this is the kind of groundwork you need for subsequent work. I mean, they're going to kind of rearrange things here, and it's kind of a big deal.
This has enormous implications for just theoretical implications for evolution and practical implications for how we think about viruses.
And also, these sequences are all available. If you want them, you could get them.
Yes. Yeah, so this happens to have been done at NCBI because that's the kind of thing they do there. But this NCBI database is totally publicly accessible.
Right. Now, I find it interesting. They make a point saying they're breaking the classification rules here in this paper because a lot of these viruses have not been formally classified, right? And that's Jens talking, I'm sure.
I could hear, yes.
Well, it's interesting. This is probably not an inappropriate time to introduce that we're talking here about phylogeny that's the sort of evolution-minded, okay, as opposed to the classification, which is, that's a taxonomic enterprise where mostly, at least initially, we're trying to group viruses by their similarities or whatever, and initially without any sort of a genome sequence. And I think one of the places this paper is going, in my mind at least, is the convergence of those two disciplines. I think that, taxonomy, I think there have been taxonomists who have been resistant to the notion of using sequence as the primary or even sole source of information for devising a taxonomy. But I think this paper is going to make a pretty strong argument for the notion that maybe that is not a bad idea.
Mm-hmm. Certainly for the viruses.
Yeah, for the viruses.
And I think last time we had Jens on, he talked about the idea that now the ICTV is moving away from needing a virus isolate to classify, right?
Mm-hmm. It has to.
Mm-hmm.
You know?
Did we define the International Committee on Taxonomy of Viruses is the ICTV?
Yes. Right.
Well, as Jens says, we classify dinosaurs, but we have no living dinosaurs.
Right.
Yeah.
And so that's the best argument. You know, we don't have any virus, but just do the sequences. So that's fine. Okay. So you take all these RDRPs and RTs, and you make this phylogenetic tree, and you get 5 major branches labeled 1 through 5. And then there's a little branch which appears to be ancestral to everything, which are the group 2 introns, the retrotransposons that have reverse transcriptase. And this is RT ancestral to retroviruses. So RTs existed before retroviruses. They just captured it.
Right.
All right. So this RT has been around a long time because, remember, the RT was required for transition from the RNA world to the DNA world. And that predated the retroviruses. So you got 5 branches. So let's go through these branches. Branch 1, leviviruses. And leviviruses include phages like MS2 and their eukaryotic relatives, viruses that infect plus-stranded RNA viruses, mitoviruses, narnoviruses, and ormeaviruses. Mitoviruses, I never heard of.
I'm glad to hear that.
Holy cow!
Me too.
Yeah.
Because I hadn't heard of half of the stuff on this.
They infect fungi and they infect the mitochondria. That's why they're called Isn't that cool?
That is way cool.
We have to do a mitovirus paper.
You get a mitovirologist on, or at least do a paper. Yeah.
Narnoviruses also infect fungi. Ormeaviruses are plant viruses, like Ormea melon virus. Yummy melons. Okay, so those are in group 1.
Somewhere along the line, he mentions a virus that may replicate in chloroplasts of plants.
Mm-hmm.
You know, I'm wondering if that's the ormeaviruses or if it was something else. I will look.
Pick it up. Look it up. Viral zone, maybe.
Well, I can search this paper for
this paper. It may be in the reference. Branch 2 is the picornavirus supergroup. A lot of plus-stranded RNA viruses like picornavirales, which is an order of the picornaviruses, nidovirales, including the coronaviruses and others, caliciviridae, potiviridae, astroviridae, solemoviridae. Okay. All right. All plus-stranded RNA viruses. And then double-stranded. Some of the double-stranded RNA viruses map in here. This includes Partiti viruses and Picobirna viruses.
And this was interesting that the double-stranded RNA viruses are not a group.
Yeah, so you'll see that a lot of them are separate, but in their own branch, but a number of them are mapping within this picornavirus supergroup, branch 2. So branch 3 is another set of plus-stranded RNA viruses, like alpha virus supergroup. That would include like Synbis virus, flavivirus supergroup, nodaviruses, tombus viruses, tomato bushy stunt virus, for example, and others that I can't pronounce. Have weird accents. I like wave virus, right? Is that it?
Wavy virus, wave viruses.
Zhao virus. Okay. Branch 4, double-stranded RNA viruses, including reoviruses, cystoviruses, double-stranded RNA viruses of bacteria. So there are not a lot of those. And totiviruses, which are viruses that infect Giardia. Branch 5, negative-stranded RNA viruses. So all the negative-stranded RNA viruses are in branch 5.
Right.
And so this is their construction, which they say is pretty strong.
And they replicated it with a couple of different tree building algorithms.
Right.
Right.
And just to reiterate again, that double-stranded RNA viruses are in two different parts of this phylogeny.
That's right.
And that's a really interesting finding. So it implies that this evolved 2 times.
So a couple of interesting features. First, they say the tree topology is compatible with the monophyly of the RDRPs, which means they had one origin, right? Mm-hmm. And by inference...
In the beginning, there was RNA-dependent RNA polymerase.
That's right.
Right.
And by inference of RNA viruses and eukaryotic RNA viruses from a prokaryotic virus ancestor. And then they say the deeper history remains murky. I love that. I think that's just great. They don't know. They don't know anything about the ancestor of these retro elements, the RT and so forth. Nothing about that at all.
The ancient ones remain hidden.
There's another aspect of this tree that I don't think we emphasized, and that is that there's sort of an evolutionary hierarchy to these branches, as if branch 1 is before.
Yes, that's right.
Branches 2 through 5.
Correct.
That branches 2 and 3 are pretty closely related to each other, but branch 3 is ancestral to branch 4, which is the double-stranded guys.
Yeah.
And branch 4 is ancestral to branch 5, which is the negative strand guys. So the implication is that positive sense RNA viruses came first,
right?
And first in prokaryotes and mitochondria and stuff, and then skipping to eukaryotes, and then double-stranded RNA viruses evolved from positive sense RNA viruses, with, of course, the weird exception of these Partiti viridae in branch 2, and then negative sense RNA viruses appear to have evolved from the double-stranded RNA virus, which is a really interesting hierarchy.
Yeah, I think that's their most surprising finding, right? They say that later on.
Yeah, that the double-stranded seems to have arisen twice, and that the minus-stranded seems to have come from the double-stranded.
Mm-hmm.
And by the way, the chloroplast viruses are in this double-stranded RNA virus branch that's in branch 2.
Mm-hmm.
Oh, okay.
And for anybody who wants to skip ahead, if you're following along in the Open Access Journal, Figure 10 has a nice diagram.
Figure 10, Figure 10 is where we're going with this. It's wonderful.
So another thing they say is the tree shows the paraphily of plus strand viruses compared to double-stranded RNA and negative strand viruses. So I think, as Rich has already said, minus-stranded viruses came from double-stranded RNA viruses. Double-stranded RNA viruses are polyphyletic, right? They're 2 different origins.
Right.
One is within the branch 2, and the other is a distinct branch. So 2 different origins of double-stranded RNA viruses. Okay, so so this is all based on the RNA-dependent RNA polymerase, but of course there are other genes in these viruses. And so they said, let's put the other genes on these viral genomes and see if we could see the gains and losses of genes over time.
Right.
And they have only one protein that they say was gained at the base of the eukaryotic virus subtree, and that is a capsid protein called the single jelly roll capsid protein.
I can hear Eugene I can't do his accent, but he rolls the jelly roll. I can just hear him talking about this. It's wonderful.
And the jelly roll, of course, is a structural element which is common in many capsid proteins, and it's ancient. And in fact, it's thought to have been stolen from a cell protein. Originally, the cell protein had these jelly roll proteins, and they were exapted as virus capsid proteins.
That's not to be confused with the jelly roll of an intestine that you section.
Right.
That's right. You remember that? Yeah, that was cool. That was very cool.
I'm thinking stolen or borrowed. What?
Yeah, they left it. They left it there. They didn't steal it. You're right. They borrowed it.
So it would be
it's
what
copyright
are jelly roll?
infringement.
Okay, but what are jelly roll proteins doing in the host? Do we know?
Yeah, that's another article by Koonin, which I don't remember, but he has a nice article where he looks at a variety of viral proteins that came from host origins, and he talks about what these jelly roll proteins do in the cell, but I don't remember at the moment.
I mean, I can make stuff up because, there's lots of spherical objects in cells. All right. There are lots of things that, might make good use of a capsid-like protein to compartmentalize stuff.
No, it's in his article. It's there, and I've blogged about it, but I don't remember. It's over a week ago. I don't remember.
Maybe we ought to check your eyes.
Right.
With the
Okay. Now, so they have, using these other proteins that they've added to the trees, they can make some gain-loss reconstructions. So here for branch 1, so they have the ancestor is a levivirus-like virus, which again these are plus-stranded viruses of bacteria. They had a capsid protein not related to this single jelly roll protein, and a certain branch came off of them, and they lost the capsid and became replicons, naked RNA replicons, and that's the mitoviruses and the narnoviruses. And they replicated mitochondria and in the cytosol. But they lost the capsid. So they came from an ancestor that had it, and then they lost it. They didn't need it. They found themselves in an environment where a capsid was not necessary. So that's very cool. So
mitoviruses are in the mitochondria, and they don't need a capsid because how do they get around?
I think they just divide with the host. They replicate, and then the host divides, and it's partitioned among the two. A lot of yeast viruses are like that as well.
Yeah.
And then narnoviruses are in the wardrobe?
Oh, you saw that Narnia thing, right? Yeah, in the big where you go in and you go to another world, right?
Right.
Yeah, they're fungi.
Okay.
Okay, so that's
basically these guys that don't have a capsid protein, they don't have an extracellular phase.
They don't have an extracellular phase.
Or even an extra organelle phase necessarily.
Right, and they came from leviviruses that did have capsids. The sequencing definitely clearly shows that. Right, they lost the capsid protein.
Yeah.
Let's see. And a group of these viruses escaped to the cytosol, and then they reacquired capsid proteins from different lineages of eukaryotic
They needed
viruses.
them again.
They needed them again. Once they escaped, they needed them. Very cool. Isn't that great?
So this is another the implication of sort of strongly implied, but not really explicitly stated, is that the evolution here, the way I think of it, is since mitochondria are supposed to be sort of trapped bacteria,
right?
Okay, that you have bacteria and bacterial viruses, and then you make eukaryotes using those bacteria along with their viruses, and then at some point those viruses decide, ooh, there's space out there.
There's a bigger world.
There's a bigger world.
Exactly.
And so the eukaryotic viruses evolve from the prokaryotic viruses via the mitochondria. Is that what the message you get?
Mm-hmm. Mm-hmm. That's right.
Wow. So cool.
Yep. Now the Picorna supergroup, branch 2, there's a lot of viruses in here. Just the Picorna virales alone have 826 members. And then there are lots of other members of non-Picornas. They're all plus-stranded viruses. And then there's this little double-stranded RNA lineage that's in this, which presumably these arose from the plus-stranded RNA viruses in this branch. A couple of interesting things here. First, hypoviruses. These are viruses of fungi. They're capsidless RNA replicons. They actually can modulate the virulence of certain fungi, like chestnut blight fungus. Its virulence is modulated by these viruses. So they've been always seen as double-stranded RNA viruses, but they say that the comparisons here suggest that they're actually derivatives of potiviruses that have lost their capsid protein. So they're not really double-stranded RNA viruses, so that's cool. And these double-stranded RNA viruses in branch 2, these are presumably viruses that took the plus-stranded RNA and just duplicated it and remained double-stranded and didn't resolve it at some point in replication. And there are some naked replicons in there as well. There's some that replicate in algal mitochondria or chloroplasts. Interesting. And they have a mitochondrial genetic code. How about that?
That's really cool.
I did not know there were viruses that multiply in mitochondria. Wow.
We need to do this.
Got to do it. We need to do that.
Mm-hmm.
They say that these double-stranded RNA viruses probably came from another prokaryotic RNA virus groups that's not yet discovered that would fit into this branch too.
That I like.
That's
that
cool.
There's a prokaryotic realm out there that we don't even know about.
Right.
Inferred from the existence of viruses.
This is really cool. It's kind of like when the periodic table was put together and could be used to predict elements that must exist.
Yeah.
Right.
So here you're putting together a phylogeny and saying, well, there's got to be some viruses here.
Right. Now, there are some other proteins that are ancestral besides this single jelly roll capsid. One of them is a serine protease that is encoded in the picornavirales genomes. And I remind you that they're called serine proteases, but they actually have a cysteine substituting for the serine in the active site. A little bit of trivia there. And this is, they think it's mitochondrial origin, this viral protease. It probably came from a bacterial protease originally, which of course then became mitochondria. So that's pretty cool. And they can see lots of gene rearrangements. But one thing I thought that was really cool is the acquisition of helicases in branch 2. All right, 3 different helicases. And these are needed, and they say here, most viruses with genomes bigger than 6 kilobases encode helicases, but the smaller ones do not. And the helicase is needed for replication of the longer genomes.
Helicases typically, they do lots of things, but one of the basic functions is that they can unwind nucleic acids. So the picture here is that when the RNA gets long, the opportunity for constraints because of the helical nature increases. Okay, and so you might need something to help sort out that the knots and stuff that might accumulate if you're trying to replicate a long double helix.
This used to be easy to explain with a telephone cord, but there aren't telephone cords anymore.
Wait, my microphone has one.
Your microphone, okay, yeah. If you have one of those spiral cords on a microphone, it gets twisted up, and then the twists get twists on them, and you get supercoiling of them, and you need to apply energy. You need to untwist that, and that's what a helicase does.
Another cool addition or change is that the virion morphologies changed. So this ancestral jelly roll protein was substituted, so the virions became filamentous in potiviruses. And in some viruses, it was replaced by a nucleocapsid protein that binds the RNA, like in the nidoviruses. All right. Another one is capping enzymes acquired by some genomes. These are needed to place the 5' cap on mRNAs, so they can trace the acquisition of those. Branch 3, this is the alpha virus and flavivirus supergroups. They say this underwent the most dramatic rearrangement in terms of gains and loss of genes. Virion architectures are different. You have capsids. You have some viruses with membrane bilayers, like the toga viruses. Some of them are flexuous and filamentous. Different capsid protein there. Rod-shaped capsids from a different protein. And they note here, which I think is very cool, the recent discovery of a virgavirus-like capsid protein in invertebrate viruses. And as an example, the Baha'i Charybdis crab virus suggests the emergence of this unique capsid fold antedates land colonization by plants at 100 million years ago. I love when they make those suggestions. 100 million years ago, you're going to, this happened before this and that. Wow, it's so cool. Here's another one. Endornoviruses are naked RNA replicons. We talked about these in a TWiV a while ago. They were originally classified as double-stranded RNA viruses, but they say here they have all the hallmarks of the alpha virus supergroup, and they're clearly derived from plus RNA viruses. They seem to have been mislabeled as double-stranded RNA viruses due to the accumulation of double-stranded RNA replication intermediates in infected cells. Isn't that cool? Well, it's not cool if you did that, but
right. Yeah.
You do the best you can with what you got when you're doing it,
Mm-hmm.
and you have to be able to accept that things change. Just actually, they got a sentence at the end of this. It's like that. It said, we aren't done yet.
Mm-hmm.
So the branch 3 we're still on. This is the flavivirus and alpha virus supergroup. They say we can't really develop a coherent evolutionary strategy for the entire branch. There are a number of scenarios for this. And we don't have enough viruses, basically, viral sequences to do this. But they say one potential clue is provided by Jingmen viruses with tetrapartite genomes, in which the protease helicase modules and the polymerases are encoded by separate genome segments. The genome architecture could hint at an ancestral flavivirus genome, which was assembled from genes borrowed from pre-existing viruses, one of which had a Tombus-like virus polymerase. I think that's right.
Back on the Tombus viruses, before we leave this group, I like the sentence where they talk about plant Tombus viruses and Tombus-like viruses with previous technology that was available. It's now changed, and they say the plant Tombus viruses now form but a small twig deep within the large assemblage that we refer to as the Tombus viruses.
But a small twig, yes.
So in talking about these changes in the protein composition of the genomes, we've been talking about acquisition and loss of genes from the host. Mm-hmm. The example that you just gave, Vincent, raises another theme, which is shuffling of genes amongst the viruses. Mm-hmm. So in terms of, building different sorts of genome compositions, what we can see from this is that you can do both. You can acquire from the host genes, or you can lose genes, and you can also swap genes with other viruses.
Mm-hmm. Mm-hmm. Branch 4 double-stranded RNA viruses, they all have they have homologous capsid proteins. They're icosahedral capsids. They keep the genome inside the capsid in cells. They're made up of 60 homo or heterodimers, T equals 1. And these include both eukaryotic and prokaryotic double-stranded RNA viruses. There are 2 branches in this family. There are 2 clades in this branch, number 4. And branch 5 is all the negative strand RNA viruses. So they're all in there and derived from, as Rich said, the double-stranded RNA viruses. Go ahead.
So one of the interesting things about the double-stranded RNA viruses, about their capsids, that's different than other viruses, is you already mentioned that the genome remains encapsulated throughout the life cycle, mm-hmm. which protects the genome. It protects the virus from detection by RNA surveillance enzymes, antiviral enzymes in the host. But one of the things they have to do that is to have all of the replication enzymes in the virus. And part of the way they accomplish that is to have a shell that incorporates a lot, if not all, of the enzymatic activities required for replication of the RNA. So it doubles as a shell and a transcription machine, and transcribes the RNA as it's pumping out. And it's also a double shell. There's an outer protein shell that's more the protective thing, and during infection you lose that, and this inner shell has all of these replication enzymes in it. So that's a highly sophisticated machine, mm-hmm. That where the capsid, I don't know the details, but I'd be interested in sort of the evolution of the structure of that capsid relative to other capsids in the other viruses. I don't know if that's, I would assume that there's a jelly roll in there somewhere, but I don't know exactly the relationship. The other thing that is pointed out is that, now remember that we're saying that the negative strand RNA viruses came from the double-stranded RNA viruses, and he makes the point that the negative strand RNA viruses, in a sense, also remain encapsulated because the RNA is in a nucleocapsid. Right. Okay, and the implication is that theme, at least, was perhaps inherited from the double-stranded RNA viruses.
Yep,
this is so cool.
Now, then they look at the single jelly roll capsid protein in some detail. And as we said, this is the one that's common, that's basal in most of the RNA viruses. It says these capsid proteins form a monophyletic group and likely were recruited from cellular single jelly roll proteins on a single occasion during the evolution of RNA viruses. So they were acquired once, and then that gave rise to all the subsequent RNA viruses that have these single jelly roll capsid proteins.
And can you imagine one time like a long, long time ago?
A long time ago.
No, it was just a really, really good idea.
So I found a paper where they say, this is Creutzfeldt and Kunin, cellular origin of the viral capsid-like bacterial microcompartments. So this is some jelly roll capsid proteins of viruses, and we show that both of them are exapted from bona fide cellular proteins, namely P2 signaling protein and OB fold domain containing protein, respectively. There you go, Rich. Those are the proteins. Cool. In bacteria, anyway.
You found that paper while you were doing all this other stuff that you're doing here? That's pretty good.
Well, you were talking, so I figured I'd take advantage of that.
Yeah, I do a little Googling on the side, too.
I know that when Rich starts, he can...
Mine is not as productive, though. So far, I've discovered that Jelly Roll, the first Google hits that you get, are a rapper.
There's a rapper called Jelly Roll?
Yeah, there's a rapper called Jelly Roll who was apparently sued by Waffle House a few years ago.
My goodness.
But
I found when I Googled for Jelly Roll and Kunin that there's a paper about double Jelly Roll.
Yes, that's right.
So that's why single and double.
That's right.
So, Vincent, according to this paper that you got here, what are the cellular proteins? What's their function?
It doesn't say. I don't know if the functions are known, but one is a signaling protein, and the other one is some domain containing. So, they don't need to be structural,
right?
And have this domain, but it was exapted to, and that happens. It has a totally different function when it's exapted.
Yep.
I like to call it copy paste.
Copy paste, yes.
They also do analysis where they ask, can we put these into groups of these various proteins that are encoded in all these genomes? Can we put them into groups based on their functions? And it's interesting that across these branches, you can see the various viruses sharing functions, and they call these modules. And mostly there's a virus family in the module, but the Reoviridae have proteins that are split into five different modules. In other words, the kinds of proteins in those encoded in those genomes are shared by lots of other types of viruses. So they make different nodes that compare that. And let's see what else here. I think that's all I want to mention, unless anyone, before we start the final summary. They talk a lot about horizontal viral gene transfer and how that might occur. Obviously, they've presented a lot of evidence for that. And they talk a lot about co-infections and vectoring of these viruses in various hosts.
And it's important from the standpoint that they think that the horizontal virus transfer is the plausible way for how these things evolved. In other words, you could imagine that that there could be coevolution of all these viruses as their hosts co-evolve, but they actually think that this horizontal viral transfer is a major part of it. Especially, they said the horizontal viral transfer by way of fungi that infect plants and insects, or that grow on. Yeah.
That's right. So multi-domain interactions, right? Like insects feeding on plants or on on mammals and fungi. So yes, that's how this happened. So they conclude that their analysis strongly suggests that HVT played an important role in the evolution of all major groups of RNA viruses, which I would say is obvious.
Plainly clear that many virus families infect a wide variety of organisms from different kingdoms, plants, fungi, bacteria, etc. So they're really moving around.
I mean, that's why you have to use a single gene like the polymerase to do this, because if the other ones have been swapped, it kind of clouds the origin, right?
Right. Right. There's too much noise on the other ones.
So let's see, a couple of conclusions. They say the derivation of minus viruses from double-stranded is arguably the most unexpected outcome, considering the lack of genes in common.
I don't think I would argue that.
Right.
Nope.
Right.
I would agree.
Yeah. And they say the flow of evolution from plus RNA to double-stranded RNA to minus makes sense in terms of the logic, right, of genome replication.
Yeah, this is my favorite paragraph. For those who are following along, it's at the bottom of page 21.
Right.
And I just love this because they start with the plus-strand RNA viruses that have the simplest strategy, and that the double-stranded RNA viruses could have evolved when a plus-strand virus encapsulated a replicative intermediate that was double-stranded along with the RDRP enzyme. And then how you can get to the naked replicons of mitoviruses, and then they say that actually isn't as easy as it used to be thought. And then, and then it's at the top of the next page that the origin of the minus strand RNA viruses, somehow the plus strand is discarded from the virions, that somehow it is evolutionarily a good thing because it simplifies transcription and replication.
So they're more involved.
Yep.
Right. I just love that whole idea of how it might have happened and rationales for it.
So I think that's it, right? I think that's a good summary.
And I think, this paper was obviously a heck of a lot of work, and I think it's going to be referenced a lot for quite a long time to come, because they've really kind of laid the groundwork for this whole new organization of the RNA viruses.
For sure, I'm going to incorporate it into my course. It makes the evolution clear. They say it's going to lead to an overhaul of the current virus taxonomy on multiple levels.
Yes, that to me is one of the biggest potential impacts on this. Okay.
Yes,
is that I see, looking at this, I see a strong justification for almost scrapping the existing taxonomy and saying, okay, we're going to do all this based on genomes. Okay.
Right.
And sure, there's a lot of other work that's gone into comparing viruses that will be useful in this, but I can see a strong argument for basing the entire taxonomy on the evolution of genomes. It makes all kinds of sense to me. And that's going to allow a grouping into higher taxons. Right now, all we have is species, genus, family, and order. And that the way the taxonomy was established was people saw viruses that looked similar, and they grouped them almost arbitrarily into the taxon of genus. Okay. And then they said, oh, there's several genera here that are similar. Let's group those together and call them families.
Right.
And they say, oh, there's several families that look similar. Let's group them together and call them orders. And now we're starting to talk about super orders and that kind of stuff. But this is going to mean rethinking it completely. And if you go back even from that, okay, I think it's time that the rest of the taxonomic world tackled the notion that viruses are either a kingdom or an empire. Okay.
Let's go with Empire.
I like Empire,
Yeah, well, I mean, that's one of the things that people talk about. The Cavalier-Smith classification scheme from 2015 has 2 empires and a total of 7 kingdoms. Okay, so there's an empire that looks like it's anuclear and one that's has nuclei, and within that there are 2 kingdoms, bacteria and archaea, in the one empire, and 5 kingdoms, protozoa, chromista, plantae, fungi, and animalia in another empire. Okay, I mean these are all just proposals. We're not sure of any of this, plus it's just people messing with it. But based on those sorts of considerations, I think the viruses are another empire.
I've always thought of them as kind of a super layer lording it over all the rest of life, that right at the very top, you just have the viruses extending across the entire classification. But that's not necessarily all that useful because there is a classification. And as this paper points out, if you can get to the evolutionary view of it, which we now can because we have the data, you come up with a useful set of classifications that tell you some things about where these viruses came from and where you might find them.
And I suppose, I don't know how the taxonomists really think, but I suppose part of the thinking is that, these empires and kingdoms are all related in some evolutionary sequence, okay? And most people, up until now at least, have probably thought about, okay, well, the viruses are just kind of out there. We can't really think about them. But another one of the implications of this paper is that we're talking about the origin of life here.
Yes.
Okay. And from that point of view, the virus kingdom is the modern representation of the most ancient form of life. Okay, and rightfully belongs in the taxonomy of living systems.
So to just summarize that, we have an RNA world, and then at some point a reverse transcriptase arises, which then leads to the production of the DNA world. But that reverse transcriptase is the ancestor of RNA-dependent RNA polymerase, which is then incorporated into genomes of these leviviruses, these phages of these plus-stranded RNA phages of bacteria, and some ancestral eukaryotic virus, which then acquires a single jelly roll capsid protein gene from the host, and then everything else has come from them.
Original viral sin.
Original viral sin. That could be a title. And so the viruses are basal to everything, for sure. There should be more than an empire. I don't know what's more than an empire. So the key, and also don't forget that as we pointed out, the shuffling of genes among not just viruses, but from host to virus, like the capsid protein and other things, is really important. It's really emphasized here. Very cool. Very cool. Mm-hmm. All right. Thank you for that.
This was awesome. This was just awesome.
Yeah. By the way, do you know who coined the phrase "the RNA world"?
No. Do you?
I do. I have the paper right here in front of me. It's a 1986 Nature commentary by Walter Gilbert.
Oh.
He says, "Origin of life, the RNA world." It's the first time it was said.
Interesting.
Yeah, I'm going to read it. It's cool. Let's do some email. Kathy, can you take the first one?
Sure. The first one is from Prasad, who writes, "Dear Vincent and all other TWiV hosts, I enjoyed the discussion on VPX-mediated regulation of Hush complex in TWiV episode 516. Jeremy's description of the whole story was spellbinding. Lanya is good too. I listened with rapt attention. May I make a suggestion? Considering that he has already been a TWiV guest 8 times, his fund of HIV knowledge, the absence of a regular HIV host on the TWiV team, all suggest that he should be invited to join the TWiV team. Not because he and I are good friends or were former co-postdocs in the same lab. Even if he were to attend once a month, that would be great. Please consider this suggestion. I wanted to recommend a great paper for discussion on TWiV. Dr. Akiko Iwasaki's recent paper in PNAS on IRV-map analysis reveals genome-wide transcription of human endogenous retroviruses. Considering the growing importance of endogenous retroviruses in our existence, parentheses, placental evolution, ARC-mediated neuronal signal transduction, and so forth, a discussion of the tools she has developed and the analysis presented in this paper is a priority. She would be a great guest. Best regards, Prasad. P.S. By the way, I am known as Prasad among family, friends, and close colleagues, not Vinny or Vinayaka.
Okay.
And so I have one comment about that, and that is that Akiko Iwasaki is going to be the keynote address speaker at ASV this summer. So
excellent.
In addition to if we discuss her paper, you can hear her present her work at that meeting.
Cool. And I actually, we saw Jeremy on Monday. He popped by the veterinary school with his lab, which was great. And we got to talking about this, how many times he's been on TWiV. I guess he's sort of to us what Steve Martin is to SNL. And I said, well, I really kind of think of you as just a rotating co-host who comes in occasionally, because he really has been on the show a lot.
Yeah, we met Lanya and Ted, who have both been on TWiV as well. So I will see if we can get Akiko to come on TWiV and discuss this paper. I have it on my list, but it seems that she would
Sounds good.
Be able to do that. That's cool. Rich, can you take the next one?
Valeria writes, Dear Twivers, it is a rather blustery day in Cambridge, UK, 10C, 50F, with occasional rains and chilly nights. We were delighted to listen to your latest podcast, number 522, where, amongst other interesting topics, you discussed our recent Nature Microbiology paper about a new ORF in enteroviruses. You had several questions and comments that we would be happy to answer. As you mentioned already, this is a new protein. No doubt it will take quite some time to fully characterize the function of this protein. One of the questions we heard not only from Vincent, but also from Eckhard Wimmer at Europic 2018 and several other virologists is why this protein was not detected before. Well, for those who studied poliovirus 2, like Vincent, it was indeed rather not possible due to the truncated UORF and technical limitations back then. It would be interesting to see now, for example, using ribosome profiling, if the short UORF in poliovirus 2 is expressed, and if so, whether it has a similar function. For poliovirus 1, the situation is not much easier due to the small size of the UP, its low expression level relative to the polyprotein, and its instability in lysates prepared from infected cells. A couple of freeze-thaw cycles, and it's gone. The dilemma on how to determine an appropriate cutoff size for defining whether or not a virus has a UP, we're saying this is upstream protein. So,
right, I think.
UP, still needs to be answered. In this study, we had to put some size constraints to perform a global bioinformatic analysis, hence the arbitrary, in quotes, cutoff length for UP. Why some poliovirus 1 sequences lack a full-length UORF also needs to be addressed with a thorough assessment of the provenance of those sequences lacking UORF. Some might be lab strains, and the point raised by Kathy about researchers correcting sequencing frameshift errors in the polyprotein ORF, but not in the presumed non-coding regions, is important. We have seen the same issue in analysis of UORFs in other viruses, such as Lutoviridae, particularly for older sequences. Another question was whether the late defect observed in infected organoids is due to a difference in the stability of the UP knockout viruses. To find the cause of the growth defect of UP mutants in infected organoids, we quantified viral protein and RNA in the late time point samples. However, even after normalizing by protein or RNA, the UP mutant titers were still below wild-type titers. This phenotype was rescued by treating the samples with detergent, which suggests that the defect is in the virus released from membranes rather than in virus stability or assembly. An interesting question which came up was, can UP be detected in virus particles? We could not detect by Western blot any UP and purified concentrated virions, so presumably it is not packaged. And finally, using this opportunity, we would like to thank our 7 awesome anonymous reviewers at a couple of journals who helped to advance and improve this story during the peer review process. That's a generous statement. Thank you, dear TWiVers, for your many years of hard work in managing such a great podcast. It is an inspiration to all in virology. Valeria and Andrew, University of Cambridge, UK.
Cool.
Very nice.
Very nice. Good. I love those kind of follow-ups. Answer our questions.
Yeah.
Yes.
Serena writes, Dear Vincent, first of all, thanks a lot for all your work with this podcast. You don't have to read my email on the show. I just have to share my frustration with you. Although my field of research, computational chemistry, is not directly connected to TWiV, I listen regularly to the podcast since 2013 as I find it highly interesting, i.e. long-term listener, first-time writer. I started listening to episode 522 on December 7 and immediately panicked when I heard you talked about flying to Zurich on the Monday after recording the show. A hasty Google search proved my biggest fear to be true. You had been in Zurich and recorded a TWiV live show on December 6th, and I had missed it. I can't believe that this really happened. In any case, I hope you enjoyed the trip to Zurich, although the weather was not that great last week, and I wish you and all the others TWiV hosts a relaxing Christmas vacation. Keep up the great work. Best regards, Serena. P.S. It's currently 2 degrees and cloudy, and this is Professor Dr. Serena Rinneker at ETH Zurich, just across town from the university, which is where I was. Well, I'm sorry you missed it.
You'll have to go back.
I have to go back.
Someday.
And if she's ever in the U.S., she needs to visit the TWiV studio. Everybody's welcome, right?
Mm-hmm. You bet. Alan, can you take that last one, please?
Sure. Stacy writes, Season's greetings, TWiVers one and all. I've been a huge fan of TWiV for many a year now, and I finally want to add my voice to the multitudes who love the weather discussions at the beginning of the program. In Bethesda, Maryland, it is currently 39 degrees Fahrenheit, 4 degrees Celsius, and the humidity is at the 90%. The morning started with gray clouds above, and it should start raining this afternoon around 3 p.m. I'm a dual U.S.-Canadian citizen, and it is a rare occasion up north when a conversation does not begin with the weather, so I could not imagine TWiV starting any other way. Please pay no attention to those voices pressuring you to change. I have followed many science podcasts, and nothing gives me as much pleasure as TWiV. It feels like listening to family. I'm including a link to a comedy skit from the Mercer Report that captures how Canadians actually survive our much harsher winters. I hope you enjoy it. Many thanks for everything I know about viruses and so much more. And Stacy sends a follow-up saying that the link didn't work, but if you type Rick Mercer, weather will be 1 degree Celsius in YouTube, you get the video, and it is pretty entertaining.
Well, when I did that, I got a bunch of videos. Do you know which one it was?
There's let's see, Rick Mercer, weather will be 1 degree was what I put in, I think.
I got like 20.
Oh, yeah, you got a whole bunch of stuff. I think it was like the first or second one that I...
Okay. We'll let people find it.
Yeah. It's the first one, RMR 7-day forecast.
Okay. Let's do some picks. Alan, what do you have for us today?
What do I have? Oh, yes. This is something somebody on Twitter pointed me toward. I think they mentioned it to you too, but I'm stealing it as a pick. It's a blog post about population mountains, and it's a way of visualizing the populations of cities. And so if you scroll down, it's a nicely designed page. The map in the background animates as you scroll. And then they plot the populations of the cities as bar graphs on top of maps. And it allows you to see things that you wouldn't see otherwise, and really kind of it is a neat way to develop a feel for the size of cities and for the degree to which they sprawl or not.
I immediately found a little peek for Ann Arbor.
There you go.
The fascinating thing is I guess you're looking, among other things, at population density here, right?
Yeah.
So there's some of these things that are these huge, thin spires.
Singapore is the most striking example, where there's just a couple little things here and there, and then there's this point just coming straight up where all the population is.
I couldn't get it to work in Safari, but it works in Chrome.
Works in Firefox too.
Yeah. So Chrome works. Yeah, that's cool. Where's Ann Arbor here? Let me see.
I don't know. I was in Safari and I found
it's because it puts you where you are. Yeah, puts you in your location. Yeah.
Oh, it does?
Yeah.
Oh, okay.
Yeah. So I'm in New York.
Well, because for me, it started with New York, Montreal, and Toronto.
Really?
But then
How do you move it over? You scroll.
I don't know.
You don't know?
I don't know.
Yeah,
but
I can't see how to move it either.
It's not moving.
It's interesting.
Yeah.
Mine is New York centered.
Yeah. It starts out New York centered, and then somehow I got it to flatten out and be the peaks, but now I can't do that in Chrome. Okay, somebody will tell us how to do that too.
Yeah,
Rich,
or Vincent, show it to your kids; they'll tell you.
They'll figure it out. Yeah, Rich, what do you have for us?
I picked an hour-long lecture called, and this, by the way, was sent to me by Ed Niles, who is becoming, has been recently and will be in the future, a major source of TWiV picks for me. A Neanderthal Perspective on Human Origins with Svante Pabo, a 2018 Nuremberg Prize for Science in the Public Interest. So this is a University of California lecture honoring Pabo with this prize for his work in ancient DNA and human origins. And we've talked about this a lot before, and I think we've had other picks in the past, but I watched this lecture and I recommend it simply as probably one of the most comprehensible summaries of his work and other work on human origins and the role of ancient DNA sequencing in figuring that out. It's really a nice and entertaining lecture, and he's an interesting guy. You know, he has a certain sort of a subtle sort of humility associated with his presentation. Just you can see it in the title, A Neanderthal Perspective on Human Origins. Okay.
Yeah, it's good stuff. Very cool. Tathy, what do you have for us?
I picked NASA space mission posters. Now, these are, it turns out NASA creates posters for every space mission, and they're hilariously awkward. And so each of the crews, has official portraits and stuff, but then they've done these and they have pop culture references in the various posters. And so this blog post has them numbered, but then within them, you can look at the particular mission number. And as I scrolled through, I saw a couple people I knew, like Chris, the Canadian, Chris Hadfield, and also the one who's the twin, the guy. Come on, guys! You know which astronaut I mean.
Yeah, I know what you mean, but I can't put a name to it.
Anyway, and his wife is married to the congressman.
Yeah,
congresswoman. Right. Anyway, Mark Kelly. Right. But I went through and I looked for one that had Astro Kate, but I didn't find one.
Yeah, that's what I'm looking for.
Yeah, so me too. Some of the themes here are great. They got one that mimics the Beatles poster with crossing the sidewalk.
Yep.
They got another one with everybody dressed in Star Trek shirts.
A lot of Star Trek and Star Wars references. Yeah.
Yes. Right.
Pilots of the Caribbean. Pilots over the Caribbean. Yeah. So anyway, they're just kind of fun to look at.
Very good.
So I just got an email, a Christmas holiday email from the Sabetti Lab, and they did this. They did sort of similar things. They put themselves in different movie posters. So I wonder if they got the idea from this or vice versa. So next week I'll make it my pick and I'll put it in. I'll put the picture in here. It's pretty cool. My pick is Mark Rober, who is an engineer who used to work for the JPL and worked on the Mars. What was that thing that walked on Mars? Not the one that's there now, but the previous
Rover?
one.
The rover?
Opportunity, Spirit.
One of those.
Yeah, one of those. I would like to know, where is he here? Anyway, the reason I'm picking him, so he had a package stolen from his porch. People get packages delivered and people steal them. So he made a package that was a glitter bomb, basically. And he's an incredible engineer. It turns out he has a YouTube channel where he documents all these things that he builds. He's an amazing engineer. I have to find out about here. This is what he did. He worked for JPL, and he worked on the Curiosity rover.
Curiosity.
Okay. And his videos are amazing. And he's got one, for example, where he made a dartboard where he's got sensors on it so that when you throw the dart, the board moves, so you always get a bullseye. He's so clever. This is the kind of person who works on these NASA projects, right? Amazing engineers. So every video, and my son last night said, yeah, I follow him. I like his videos. He does all kinds of cool things. And so the glitter bomb trap is cool, and the automatic dartboard is also cool. Oh gosh, it's so creative, the stuff that he can do. And he's got millions of followers because he's very creative, of course.
It's a great video. My brother had sent me this too, and I didn't look at it till you put it in the pics.
The glitter bomb trap?
The glitter bomb. It's amazing. And then he kind of beats himself up over it in the comments because he included a couple of things that where the reactions weren't real, but he's since deleted those. But anyway, I wanted to comment to him and say, don't beat yourself up. This is a really great thing. Just the engineering of it. And so in addition to including the glitter, he's got 4 cameras to photograph every angle so it catches the perpetrator's face. Yeah. It's got a stink bomb aspect to it that sprays out this horrible smelling stuff. And so it contaminates the car or wherever you open it up. And it's pretty amazing.
Yeah, don't mess with a NASA engineer. We have a listener pick from Neil who writes, hi, Vincent and TWivers. If you have time for a listener pick, I'm finding that I check Snopes.com more and more these days for comfort. Here's an example. Fact check. I think you picked this originally, Rich, right? Snopes.
Yeah, some time ago.
Was a scientist jailed after discovering a deadly virus delivered through vaccines? Claim. Researcher Judy Mikovits was thrown in jail after she refused to discredit research that led to the discovery of retroviruses transmitted through vaccines. And this is, of course, not true.
No.
She was jailed because she wouldn't show her give prosecutors her lab notebooks, or someone her lab notebooks, right? She was involved in the XMRV CFS business.
Boy, that brings up, actually deep emotions from long ago on TWiV.
Mm-hmm. Yes.
What a mess that was.
It was a mess, but it got sorted out, right?
Yeah.
Mm-hmm.
Trust science, not scientists. Thank you, Neil. Yeah. All right, that's TWiV 526, microbe.tv/TWiV, or any podcast player on your phone or tablet. Please subscribe so that we know how many people are listening. I know a lot of you don't like to do that because I don't know why. Some of you say it'll fill your phone up, but it won't. Most podcast players stop at 2 or 3 episodes and they discard the older ones. So
and you can set the settings.
Yes, too. And so there's no downside. Just subscribe, and that will help us. If you really like us, give us some contributions over at microbe.tv/contribute. And of course, your questions and comments go to TWiV at microbe.tv. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Thank you. This was a particularly fun one. This is great.
I was
always
worried,
a good time.
I was worried you guys wouldn't particularly.
when I looked at that, and I thought, oh my God, I'm gonna deal with it. And then I got into that paper, and just all kinds of stuff came up. It was great.
It's cool. Speaking of emeritus, I tweeted something today, and someone retweeted it. Emeritus Professor Vincent Racaniello at Columbia University is asking blah, blah, blah. I said, wow, why did I become emeritus? Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.